Hi everyone, I'm Zeynep and I'm a postdoctoral researcher in University of Évora in Portugal. I work on factor shaping trophic interactions in freshwater food webs. And today I will talk about my postdoctoral project, which is about size abundance scaling across biogeographical gradients in aquatic food webs. First of all, I would like to give you a brief example of an aquatic food web. In the base of the food web, we have phytoplankton, which are primary producers. And on the second level, we have zooplankton, which feeds on phytoplankton. On the third level, we have macroinvertebrates, which can feed on zooplankton and phytoplankton. And on the fourth level, we may have fish, but not in all aquatic food webs. In nature, these trophic interactions are very complex and dynamic. To unravel the food web dynamics, body size can be used as a synthetic index, especially in aquatic ecosystems, because most aquatic ecosystems have indeterminate growth. So they differ in size as they develop, and they also shift their niche, and this creates size structure communities in these ecosystems. Body size is also important because most biological rates, such as growth and metabolism, are highly dependent on body size. Size influences predator and prey interactions because predation is size dependent, and size represents a trade-off between population growth and susceptibility to predation. So body size can give a useful information about food web structure. Size spectrum is another concept that can be used to infer ecosystem functioning and energy transfer in aquatic ecosystems. And this goes back to 1970s when it was mostly used in marine ecosystems, but later it's also used in other aquatic ecosystems and even in terrestrial ecosystems. It can be also defined as size abundance scaling, and it's a linear regression between body mass and abundance in logarithmic scale. The slope of this relationship gives a relative estimate of distribution of large and small organisms in the community. As you can see here, this relationship holds true for single trophic levels, for phytoplankton to fish and mammals, but it's also true for across multiple trophic levels. And this relationship can also be used as an indicator for environmental disturbances, such as pollution and climate warming. I would like to give you some examples for these environmental disturbances. For example, in this experimental study, they found that the slope of this relationship increased with warming in phytoplankton communities. So with warming, smaller phytoplankton increased in abundance while larger phytoplankton decreased. In another study for macroinvertebrates, they found that in polluted streams, this slope of this relationship decreased, where they have smaller abundance of smaller macroinvertebrates and larger abundance of larger macroinvertebrates. However, not many studies have incorporated size abundance scaling of several trophic levels across different environmental or climatic gradients. So by using Iberian pond network in the Iberian Peninsula across biogeographical regions, we wanted to assess changes in size abundance scaling and thus in aquatic food webs across regions. This experimental facility comprises 6 locations, 4 in Spain and 2 in Portugal. And these locations cover a variety of climates, ranging from semi-arid to alpine to temperate and to Mediterranean. In these 6 sites, we have 32 enclosures, and then we have data for 3 years from 2016 to 2018. We also sample environmental variables and food web components, which are phytoplankton, zooplankton, and macroinvertebrates. We have taxonomy and abundance data from these 3 trophic levels. We expect to see some changes in size spectrum across regions. If only one functional group responds to an environmental factor, we expect to see change in the slope of this relationship. And if all groups respond in the same way to this environmental factor, we would observe change in the intercept of this relationship. We may also observe that different functional groups respond in a different way to this environmental factor, and then we would expect to see changes in slope and intercept of this relationship. So for Iberian ponds, we hypothesized that we would expect steeper slopes and increased intercepts with the increase in temperature, because we expected that small organisms increase while the large ones decrease in abundance with the increase in temperature. Here you can see the abundance size spectrum for Iberian ponds. Different colors represent different sites, and we also have different groups of trophic levels from phytoplankton to macroinvertebrates. The black line represents the slope at steady state conditions. And as you can see, all sites differ from the steady state conditions, so they all had an impact. However, we also see that there are some differences between sites. For example, Evora and Murcia, which are the yellow and the dark red, they were different from the other four sites. And these are the places that have the higher mean temperature. And so these sites had smaller abundance in smaller size classes and larger abundance in large size classes. And when we plot the slope and intercept of this relationship, we also observe the same, that the Evora and Murcia were different from the other four sites in their slope and abundance. And there were also some differences between other sites in terms of their intercept. And when we conduct the linear mixed models using ponds, mesocosms as random factor in each site, and site, body mass, temperature, and site as predictors, we observed that there were significant site body mass, temperature body mass, and site year interactions. So this indicates that this relationship changes across sites with temperature, and there were some differences between years in each site. So these are still some preliminary results. We still continue to analyze other factors such as drought and precipitation that could have affected these observed patterns. And these results were based on the first part of the experiment, where we had the reference conditions without any treatments for these 3 years. However, starting from 2019, we also had some treatments such as warming and drought conditions. So for the following years, we will also incorporate these data to understand how warming and drought conditions also change these relationships in aquatic ecosystems. So thanks for your attention, and I would like to thank to my colleagues and the funding from several funding agencies. And thanks for joining us for the International Humboldt Day. You can see more events and videos in this link. Thanks again.